Welcome back friends, welcome to another lecture from Just Biology. In this lecture we want to talk about a replication process in both eukaryotes as well as the prokaryotic cells. Before starting the lecture we want to discuss something regarding the cell division, then we'll move on to the replication process. We know that they are the complex process which takes place inside the cells. So that isn't it. If you look at the structure of the genome of prokaryotes as well as the eukaryotes, we could find many dissimilarities between them. And in this lecture, I'm not going to talking about dissimilarity between those two things. But though I want to talk about entire process like cell division and how this process will begins and how the cell will committed into the cell replication. Then actually, what you mean by replication? The replication is a process by which the DNA gets multiplied, isn't it? So this is the process of DNA replication. Like if you see the entire process inside the cell, so that is, just imagine the cell cycle. Cell cycle, we have different stages. If you take interphase, mitotic stage and so on, so that is too specific, and each and every process will be specific at a specific stages. So that is very important here. Now, so I want to talk about like G0 phase, G1, then S phase. And why? Because this replication process will be starts at G1 phase and that will be end at the S0 phase, S phase. So this is a scenario when we talk about a replication. Now as the process will be going on like inside the cell, so we are not able to distinguish whether they are in different kind of stages or something. So this process will be takes place in a while. So for us to easy to understand this process, we will classify this process into three different stages. I'll tell you. The first stage is the initiation process. It's the initiation. The second phase of the replication is elongation. Third phase of replication is termination. So I'll tell you each and every process in a moment later. So before, we should know the cell cycle of both eukaryotes as well as the prokaryotes and what makes difference between them. If you see the prokaryotic cells, they have no specific stages like G0, G1 or something like that. But in case of eukaryotes, they does the stage like G0, G1, S phase, M phase, then again G0. So this is the scenario of eukaryotic stage. When we look at the prokaryotes, they have no any kinds of these stages like G0 or G1 or so on. Now, then usually the bacterial cells. So when we talk about bacterial cells, we should take an example like, so let us take an example E. coli. We'll take E. coli for this experiment or the replication. Now, if you see the E. coli cells, they have they undergo a specific cell division, unlike eukaryotes. I'll tell you. So normally the E. coli cell will go a process of cell division by a specific process. We can call it as binary fission. We can call it as a binary fission. Binary fission means the cell will get multiplied or divided themselves by increasing their some physical properties of cells. So that is what we'll discuss now. Then we'll move on to each and every steps in detail in upcoming lectures. Now, as I said before, the bacteria will possess a specific process. We can call it as binary fission. Let's move on to the binary fission. So let us consider this is an E. coli cells. So they have a genome. So inside the cell you can see a cytoplasm, and some subcellular organelle and so on. You can see when you analyze the bacterial cells. So as far as the cell concerned, there is a ratio between nuclear content and the cytoplasm. We can call it as nuclear content is to cytoplasmic ratio. So usually the cell will maintain a specific ratio when we talk about, so when the cell is in a normal condition. But during this binary fission, it will change the content ratio. The nuclear content will become less as well as the cytoplasmic content will become more, so that they can easily make the gap or make the space for the initiation, elongation and termination as well. So that is the idea. So here we have this cell. The cell will slowly increase their cytoplasmic content. How? So they will slightly increase their cells or enlarge their cells. So that's why, so they are able to multiply inside this cell. So that is the idea. So this is the genomic content, and the rest you can see the cytoplasmic content. So this is the idea. So once they made enlarge their cells or by themselves, now what should be happen next? So the cell will committed into the binary fission. We should know the cell have a certain property or certain physical nature. As I said before, the content ratio should be same. But the cell, if they are enlarging these kind of things, what do you say, the content ratio, cytoplasmic or cytosol ratio, so that means thereby the induction of the signal of binary fission. So that is the idea. So they will create a signal for the multiplication. So they will create a synthesis. They will create a signal for the multiplication by the binary fission, so that they can easily divide themselves. Well, now you can have the question, where does the replication is actually takes place, or which stage or which part of the cells you can see the replication process. So I'll tell you. Actually, the replication process will happen during the cell enlargement stages. So that is, the cell again will possess a bigger size. Also that would become a little big than as it's supposed to be previous stages. Now the content will be undergoing, or they are committed to replication, and the new daughter molecule or the daughter DNA will be synthesized. So this is the idea. So it's supposed to synthesize a next generation, that is, actually this is the parental DNA. The parental DNA will committed into the replication. The replication will give you the entire or the whole content or duplicated content here. Now the cell will have a 2 DNA or 2 genomes, as it's supposed to be, it's supposed to be cleaved off, and it should become a 2 cells. That is the idea. The binary fission will give you the 2 cells after the initiation, elongation and the termination process will happen. So once the cell will finish all this process, you will get 2 things. How? Once the last stage of the replication, that is termination, is finished. Now the cell started to making a wall between two genome here. So the wall between 2 genome here. So we can call this wall or the barrier, we can call it as septum. The septum means a barrier between 2 genome. So this is the 2 genome. The barrier between 2 genome, we can call it as septum. So this septum will disintegrate, or the septum will take each cell apart. So that is the idea, and it would become 2 cells. So that is the idea. So it would become 2 cells with the same information which supposed to carry. So these 2 genetic material, this information should be same, or actually more or less both are same. And the further these two cells again undergo the same process. So that is the idea. The binary fission, the cell will get enlarged the once the cell, the content, the cytoplasmic or the nuclear content ratio will increase, then they started to synthesizing the DNA by the replication process. Then once they finish the replication process, the wall of the a wall will be formed or the barrier will be formed. As the result of this wall formation, the cell would become two different cells. The two different cells will further undergo the same process. As the results, we will get a many number of bacterial cells. So this is regarding the normal bacterial division in that belongs to the prokaryotes. Now in upcoming lecture, that is 3 consecutive lecture I am going to take here. The first lecture would be initiation, the second lecture would be elongation, the third would be termination process in detail. I hope this content is enough for you to understand the basics of replication in prokaryotes. Now I will move on to the next initiation, then elongation, then termination. I hope you like the video. If you like the video, don't forget to subscribe my channel. If you like, definitely please share with your friends.